Hello friends, today's topic of our discussion is Selaginella life cycle. Here, Selaginella life cycle we have divided in two parts. Part first, today in part first, we try to complete classification, number of species, then its distribution, then sporophytic plant body structure, then anatomy of the sporophytic structure and vegetative reproduction. And in next lecture, in part second, we will try to cover the spore producing structures, okay, strobili, then megasporophyll, microsporophyll, their sporangia, their spores, their germination, gametophytic structures, and alternation of generation. So today we are starting with the Selaginella life cycle part first. Systematic position. It is belong to division Pteridophyta, subdivision Lycopsida, order Selaginellales, family Selaginellacea, and genus is the Selaginella. Okay, this is about the classification of Selaginella. Then there are about 700 species of Selaginella cosmopolitan in distribution. There are total number of species of Selaginella is 700 and its distribution is cosmopolitan, means they can occur everywhere their life of the Selaginella is possible. Number 2, they are commonly known as spike moss or small club moss. Due to their body appearance, they considered as spike moss or small club moss. Number 3, most of the species inhabit damp and shaded forest of tropics, but some grow in xerophytic habitats. As they are pteridophytes, they are growing in the shaded forests in the tropical regions, but some Selaginella species also shows the xerophytic nature. Number 4, the genus is represented in India by more than 70 species. This is the total number of species in India. Among these, Selaginella kraussiana, Selaginella monospora, Selaginella biformis, Selaginella rupestris, Selaginella megaphylla, Selaginella bryopteris, Selaginella ciliaris, Selaginella chrysorrhozoides, Selaginella pentagona. These are the common Indian species. Now the sporophytic plant body. Sporophytic plant body means spore producing plant body, which is the dominant plant body in the pteridophytes. Long lived plant body in the pteridophytes. It is the typically green for nutritionally independent plant body in the pteridophytes. Okay. Now the point number 1. The prostrate stem is dorsiventral. The stem of the Selaginella is dorsiventral, means it is somewhat flat, shows the upper dorsal lower ventral side. Number 2, leaves are arranged in pairs. You see in the photograph or in figure, leaves are always in pairs. Each pair consists of two different sized leaves. You see in the pair one size of the leaves are the different. One is small, another one is the large. Number 3, the large leaf of each pair is inserted on the ventral side of the axis. The large leaves on the axis produces at the ventral side, back side, lower side of the stem. Number 4, the pairs of leaves are arranged in such a way that the small leaf alternates the large leaf. Due to their arrangement, small leaf alternate with larger, larger leaf alternate with the smaller. So this is the arrangement of leaves on the stem. Number 5, leaves are quite small in size, somewhat triangular or lanceolate. They are small, triangular in shape, or lanceolate means long in nature. Number 6, the leaves may be thin or moist loving species. The leaves are very thin when the species are grows in the moist conditions and thick in xerophytic species. When the species of Selaginella grows in the xerophytic conditions, at that time leaves are the thick. Number 7. The prostrate stem bears a elongate, colorless, leafless, cylindrical, downwardly growing structure called as rhizophore. Okay, the rhizophore structure is developing from the main axis or the stem, which is the long, colorless, leafless, cylinder-like, cylindrical, and grows inside the soil. And this structure is called as the rhizophore. Number 8. At the terminal means distal end. At the terminal end, the rhizophore, a tough top adventitious roots developed. This rhizophore produces the adventitious roots at their tip. Okay. Number 9. The adventitious roots are thin. These roots are thin, slender, okay, then delicate and dichotomously branched. These leaves are thin, slender, delicate and shows the dichotomous branching. Number 10. The reproductive structures are formed at the apices of the branches. Okay, you see in the diagram at the tip of the each branches shows the compact masses of the leaves. That compact masses of the leaves are nothing but the spore producing structures called as the strobili. Okay, so this is about the diagram about the Selaginella habit. The leaves are there, small leaves, larger leaves. Okay, the stem, then stem produces the rhizophore. Rhizophore again produces the dichotomous branching roots. They are adventitious roots. Okay, then stem is the branched, etc. These are characteristic of the habit of the Selaginella. Now the anatomy means internal structure of the sporophytic plant body. First anatomy is the transverse section of stem, means anatomy of the stem. Okay. Number 1. The epidermis is prosenkymatous and cutinized. Okay. Epidermis is single layer which is prosenkymatous and which is further covered by the cuticle. Number 2. Just beneath the epidermis, the cortical region is found. In some species, the cortex is composed of thin-walled parenchymatous cells having no intercellular spaces. Okay, just beneath the epidermis, there is a composed of parenchymatous cells. Okay, and this parenchymatous region is multilayered called as the hypodermis. Okay, majority of species possesses rigid stems, have compact sclerenchyma in the hypodermal region beneath the epidermis. In some species in the hypodermis, just below the epidermis, there is a patches of the layer of the sclerenchymatous cells. As they are mechanical, they give rigidity to the stem. Okay, generally it happens in the xerophytic Selaginella plants or Selaginella species. Number 3. You see in the diagram, epidermis, beneath the epidermis, hypodermis is there. Beneath the hypodermis, cortex is there. Okay. Number 3. Endodermal cells are stretched. Endodermal cells are below the cortex. There is endodermal cells. Endodermis is always separate the inner stellar region and outer extrastellar region from each other. Okay. And here endodermal cells are not single layered. Again they produce the stretched structures called as the tuberculi. Okay. Endodermal cells are stretched, having large intercellular spaces. In between the tuberculi, two tuberculi, there is a intercellular spaces. Okay. Number 4. The stele is simple protostele to a complex polycyclic siphonostele. And here still the stellar region is very simple. which is the polycyclic. Okay, then siphonosteel. Then number 5, the steel remains surrounded by trabecular endodermis. Just you have seen in the diagram, at the center steel is there and steel is further covered by these tuberculi endodermis, okay, or stretched cells of the endodermis. Number 6, each steel possesses there are two steels in the uh, single stem anatomy. Okay. Number 6, each steel possesses a single mass of exarch protoxylem in it. Okay, and here in the in case of the xylem, the protoxylem is exarch. The protoxylem is composed of annual and spiral tracheids. The tracheids are present in the protoxylem, and the tracheids are having the annular and spiral thickening. Okay. Number 7, the metaxylem in each bundle is centrally placed. The metaxylem in each bundle is centrally placed and lies centripetal to the protoxylem. Okay. The metaxylem is the composed of scalariform tracheids. Centrally placed metaxylem is there. Metaxylem is nothing but the larger cavity xylem cells, and they are made by the scalariform tracheids. Tracheids are there, and tracheids are further characterized as with scalariform thickening. Okay. Number 8, centrally located xylem of the steel is ensheathed by a few layered phloem. Okay, so xylem is centrally located, which is further ensheathed by, covered by the few layer of phloem. The phloem is composed of sieve tubes, companion cells. Okay, the phloem is composed of sieve tubes, and sieve tubes one of the elements of the phloem. Here the phloem contain sieve tubes and companion cells are absent. Okay. Number 9, each steel remains surrounded by a single layered pericycle. Okay, below the endodermis there is a pericycle, and pericycle is single layered, and each steel is further covered by the pericycle. In this way, this is the anatomy, internal organization of the stem. The organization is somewhat complicated, but if you go through the details, or if you see the diagram, then you will read the explanation, then you will be very easy to understand the anatomy of the stem. Then the anatomy of the leaf. Okay. Number 1, both the upper and lower epidermis of the leaves are distinct. See in the diagram, upper epidermis is single layered, lower epidermis is single layered, both are separate from each other. Number 2, the epidermal cells have chloroplast. These epidermal cells contain the chloroplast, so that they are involved in the photosynthesis. Number 3, stomata are distributed mostly in the midrib region. At the leaves are having the single midrib at the center, and at the central region only stomatas are there. Number 4, the mesophil means tissue present in between the two epidermis, upper and lower epidermis. The mesophil consists of loosely arranged thin-walled cells with many small or large intercellular spaces. Here mesophil contains loosely arranged cells with small or large intercellular spaces. These intercellular spaces are easily seen in the diagram. It is usually made up of spongy parenchyma, and this mesophil is made by the spongy parenchyma. Number 5, spongy means sponge-like. Number 5, a mesophil cell have 1 to 8 cup-shaped chloroplasts. Okay, each cell having the 1 to 8 cup-shaped chloroplasts, which have many spindle-shaped pyrenoid-like bodies, and each cell again having the spindle-shaped pyrenoid, which is the starch synthesizing body. Number 6. The leaf has a median vascular bundle. Median means centrally located at the position of the midrib, surrounded by a distinct bundle sheath, and this centrally located vascular bundle further covered by the bundle sheath. Number 7. The xylem, which occupies the central part of the bundle. At the center of the vascular bundle, xylem is there, consists of only tracheids with annular or spiral thickening, and this xylem contain only tracheids having the annular and spiral thickening on them, and further this xylem is covered by the phloem. So in this way, this is the very easy to understand the internal organization of Selaginella leaves. Okay. Now at maturity of the plant body, the Selaginella sporophytic structure goes in the vegetative reproduction. Okay. First point, vegetative reproduction in Selaginella takes place by tubers, bulbils, dormant buds, and by fragmentation. Okay. So these are the four types or organs of vegetative reproduction: tubers, bulbils, dormant buds, and fragmentation, which is the most common vegetative reproduction in cryptogams. Number 2. In Selaginella rupestris, prostrate branches produce roots during favorable conditions. Okay. In this species Selaginella rupestris, prostrate branches produce roots during favorable conditions. These root-bearing prostrate branches separate from the parent plant and grow into new sporophytic plant body of Selaginella. Okay, these prostrate branches separate from the parent plant and causes the vegetative reproduction. So this is the vegetative reproduction caused by the prostrate branches. Number 3. Species like Selaginella chrysorhizos and Selaginella chrysocaulus propagate with the help of tubers and bulbils. These tubers and bulbils are developed in these two species. The tubers may be aerial, they will develop on the aerial branches. Okay, developing at the apices of the aerial branches of subterranean. They are developed at subterranean position or aerial position. During favorable conditions, the tuber germinate into new sporophyte. Okay, and this tuber causes the vegetative reproduction in these two species. Last one, aerial branches of Selaginella chrysocaulus also bear some dormant buds. Okay, these species causes some dormant buds, okay, which grow into new plants during favorable conditions, and these aerial branches producing buds in dormant condition, they produces the buds, and these buds further causes the vegetative reproduction, means they again produce the new sporophytic plant body of the Selaginella. So in this way, these are the vegetative reproduction in Selaginella takes place by different ways. Okay, so in this way, here vegetative reproduction is over. Now we can stop, and in next lecture we will observe the strobilus and its further details. Thank you.